chapter 1. So I mentioned earlier in the first part about this genus and species name of bacteria such as E. coli. So this naming system or nomenclature was first established by Linnaeus in 1735. And he came up with having this two name system, genus, and we tend to say species name, but it's really the formal name is specific epithet. And this was also in Latin, and that's because that was the language of scholars at the time. We always capitalize the genus, and so I did that here, right? Genus is capital, and the specific epithet or species name is lowercase. And it's very important that you follow this when you write, that you capitalize the genus name in lowercase for the species. Now, when you're writing on the computer, typing, you would put the entire name in italics. If you were handwriting, you would underline it. And once you've spelled out the entire genus and species name the first time you use it in a paragraph or a paper, whatever, after you've written it out fully, then you are allowed to abbreviate it after that. A proper abbreviation is only an abbreviation of the genus name. So, for example, Escherichia coli thereafter can be called E. coli, and notice that they are still in italics. Why did he come up with the system? Because scientists all over the world need to be able to communicate with each other, so everyone needs to be able to use the same name for the same organism, or else it would greatly slow down progress. Sometimes you'll see the genus and or the species name is describing a characteristic of the organism. That could be referring to the location that's found in the body, the location that's found on Earth. It could be describing the disease or one of the symptoms of disease, or it could be honoring the researchers that discovered it, right? So you'll see variation. Sometimes the names are very helpful in telling you what telling you some information about the organism, and other times it's not. Okay, so I thought I hit the next button, but I guess I didn't. Okay, so classification is the organization of all life and figuring out which organisms are closely, we say closely related or very similar to one another, and how similar. And you can make this very elaborate branching diagram to include all the organisms. I've included the most simplified tree to demonstrate that to you here, which is the current system. But when they first started doing this organization, this classification of living organisms, it was just a very simple, okay, we know we have plants and we know we have animals, and they're quite different from each other. And there's going to be other organisms, but we don't really know. We're discovering what they are and how they fit into this view. And so that was the first tree. And then after that, there were lots of different, more and more complex trees made as we discovered more organisms, as we got better tools to learn about organisms. And so now today we have this 3-domain tree, right? So we know there's bacteria, and you're going to have, billions of different types of bacteria. Then you have archaea, which are also single-celled prokaryotes, meaning they don't have a nucleus, but they are very different from bacteria. So you can see that they're different because they are far away on this tree. They are their own group of organisms. And we won't talk a whole lot about archaea throughout the class because they don't cause disease. So let me go ahead and give you a little information about archaea right now. It might come up, here and there a couple times. You'll learn maybe a little bit more about them, but this will be the bulk of what you learn about archaea right now. So make sure you're taking notes. Of course, every lecture I have some information written on the slides, and then I give you a lot of additional information that you are responsible for knowing. The nice thing for you guys is that it's recorded. You can go back and fill in any gaps in your notes, which you wouldn't be able to do in person. Okay, archaea bacteria, diverse group of prokaryotes. As I mentioned, they are very different from bacteria, but they are still prokaryotic, meaning they do not have a nucleus. They are still considered a simple type of cell. And if they have a cell wall, that cell wall is not going to be composed of the same chemical structure that bacterial cell walls are made of. And we have found that through looking at their RNA, that they are very different from bacteria, which is why they're so far away on this tree. And even their plasma membrane is a bit different. So, looking at them close up, looking at their molecular makeup, they are very different from bacteria. We have three major groups of archaeobacteria, and those are methanogens because they produce methane, halophiles because they live on or in a community with very high salt content, and then thermophiles because they live in very hot environments. Right, so the methanogens are anaerobic, meaning they do not need oxygen, and their waste product is methane. They can be found in sewage, right, or like a bog, and even in the intestinal tracts of ruminants like cows. Ancient methanogens are the source of natural gas, right, methane gas. Halophiles, as I mentioned, thrive in high salt concentrations, such as a salt lake or pool or seawater, right? So high salt concentration. And then thermophiles, like I said, live in hot environments. So thermophile means heat-loving, right? So heat-loving bacteria found near things like hydrothermal vents where you might find some hot springs if it opens up on the top, right? Many thermophiles are also have their own interesting type of metabolism called chemosynthetic, meaning they don't eat food like we do, or even use the sun. They don't use right. They're not photosynthetic. They're chemosynthetic. They're using dissolved things like dissolved sulfur and other elements as their energy source. So this is also very unique. Okay, so that's it for now about the archaeobacteria. And then our third group, the eukaryotic organisms, which are broken into further subgroups, which we'll see in a moment. But eukaryotes have a nucleus. They're a much more complex type of cell. They have organelles, right? Like the nucleus is an organelle. And then they also have things like the endoplasmic reticulum, right? The Golgi apparatus, all those parts of the cell that you've learned in the past, and you'll see again in the next, in a couple chapters. Okay, so eukaryotes broken into, so eukaryotes are a domain, archaea are a domain, and bacteria are a domain. So the three domains of life: bacteria, archaea, and eukarya. But eukaryotes can be further broken down, and we'll come back to this in a second, Oh, I deleted that slide. That's right, I forgot. So eukaryotes can be further broken down from domain into kingdoms, and those kingdoms are you have the plant kingdom, the animal kingdom, fungi, protozoa. There's actually a lot more kingdoms as well, but these are the main kingdoms that we see in this class. Okay. And so how do you work with this naming system? So it is a 3-domain tree, but within domains you can have further and further subcategories until you get all the way down to that genus and species name. And so the example of that classification system, or the hierarchy, right, is a domain, kingdom, phylum, class, order, family, genus, and species. And see how it gets smaller and smaller. So every eukaryotic cell-based life will fit into the domain, right? So that's a huge group, billions of organisms in this group. Then you'll have different kingdoms. So some of the organisms in this domain will go into the Animalia kingdom, but others will fit into the other kingdoms like plants, fungi, etc. And within this animal kingdom, some of those animals will be in the phylum Chordata, right? They have a spinal cord. And then some of those organisms will fit into this class of being a mammal, right? And so on and so forth until you get down to the exact organism that you're interested in this case, the red fox. And so this just shows you a visual of this hierarchy, this naming system. I used to, once you repeat this enough times, you'll have it memorized. But when I was a student like you, I used this mnemonic to remember the order of these. And so I say, do keep pots clean or families get sick. And the D is for domain, K is for kingdom, P is for phylum, C is for class, order, family is actually family, that's kind of nice, genus, and species. So hopefully that's helpful to you. Okay, so what about viruses? They're not living and you can't see them under a microscope. They always have to infect a cell to reproduce. And so we call them obligate parasites. They have to infect a cell to reproduce. Viruses are classified by their genetic material. Is it DNA? Is it RNA? Is it one strand or two strands? See, our DNA is double-stranded, right? We have the double helix. But viruses can be single or double-stranded, and it could be DNA or RNA as their genetic material. So that's kind of the first category for classifying viruses. And then you'll notice these shapes are very different, right? So their physical characteristic, does it have spikes? Is it round? Is it, this shape here is a bacteriophage, which only infect bacteria. And I just think they're so cool. This picture, I remember seeing a similar picture like this when I was a student, and it really fascinated me, and that might be part of why I went into microbiology, and I also work with viruses in my research. So they look like little aliens that have landed. And they inject their DNA directly into the bacterial cell, which is just so weird. It's so different than what we see with animal viruses. Anyways, we'll have an entire chapter devoted to viruses, so I will move on a little bit. But one thing I want you to start thinking about is that viruses are very specific. You only, like, one specific virus will have a very few types of cells, sometimes even just one type of cell that it's able to infect. This is why, you never worry about touching plants and getting sick. Like, you don't worry about your hands getting infected from touching plants and animals, right? This is why you can be sick with a cold and your cat or dog doesn't get sick. these viruses, not all illnesses are viruses, but those that are going to be host specific. Occasionally, we come across a virus that does cross the species barrier, and then we become very concerned. So you might remember all the panic over influenza, when we thought that like the swine flu, the strains that could go from pigs to humans, or the avian flu from birds to humans, right? And these are the reason that these are so scary is because this is not how it usually works. And when you get a virus that can go from animals to people, you now have a much more difficult case of trying to contain, but you also have something that's just different than what you're used to working with. And so there's always that danger that it could be more deadly or more difficult to treat, right? Things like that. Okay. Now we will start the remainder of the lecture will be on the overview of the history of microbiology. And we will talk about a lot of different people, but of course there are so many more that we're not talking about. So just remember that each scientist really builds upon the work of other scientists. So as much as we might put these people up on a pedestal for their contributions, they didn't do this in and of themselves. They used other people's information and built upon that information. Your book talks a little bit more. Make sure you're reading your book. Okay. And also I want to say, because I don't know if I've said this yet, there's quite a bit of information in my lectures that don't come from your book. So you'll want to read your book. It'll help explain the concepts. It'll have diagrams and things. It's really great. It has practice questions at the end of the book. But make sure you're also taking notes during the lectures, because there will be information that's not found in your textbook. Anything I say in lecture, you are accountable for knowing. If there's something in your textbook that I did not mention at all in any way, then you don't need to know that. I don't put random tests, questions just to see if you are doing your reading. Although there might be something that was explained much more in your book and I only had time to mention it, and then that is fair game to put on a test. Okay, so Robert Hooke, he's English architect. In this day, science, very early days of science, scientists had to have a job to support their interest and investigations into science. And so he was an architect by day, physicist and philosopher by night, you could think of. And he actually made a simple microscope, and then he recorded everything that he saw under the microscope, and that some of those sketches and descriptions were put together in a book and published, and it's called Micrographia. He improved upon what was already a simple compound microscope at that time, so he was able to have some better images and better sketches. And so we really started to discover and learn about this new world of organisms that exist that are smaller than anything you can see with the naked eye. And this really is a huge new concept. Nobody knew that life existed beyond what you could see, beyond what seemed tangible, right? So no one knew that living things smaller than the eye could see even existed until these improved microscopes came about, and we could actually start to see these small little things crawling around. So up until this point, people believed that life, like things like flies and all of life, basically just was kind of a magical, mystical force in the air that they called the vital force that would just make life appear, because they didn't know what microbes were. Okay. Um, so what did Robert Hooke do? What is he known for doing? Well, he sliced a piece of cork and looked at it under the microscope and saw these repeating box units, which he called cells. It reminded him of cells of a monastery. Um, and that really is the beginning of microbiology. Of course, those cork slices dead plant cells, but that's still the first cell that is recorded. Okay, then Anton van Leeuwenhoek, is Dutch, tradesman by day, scientist by night. He further investigated, using a improved compound microscope, and he observed living microorganisms. He looked at samples like rainwater, feces, plaque, anything he could think of, and he, like I said, he made even more improvements with improved resolution. And so Anton van Leeuwenhoek is really the first one to describe living microorganisms, and his work really challenged this current idea of spontaneous generation. It's his work that really set the stage for everything that we're going to talk about today. We might have to do this in a third, video as well. Break this up. Okay, so Anton van Leeuwenhoek was very good at making microscopes. He made lots of improved resolution microscopes and did a lot of work with this. One thing I forgot to mention with this micrographia, he did a lot of looking at plants and things up close, whereas Anton van Leeuwenhoek, as I mentioned, he looked at a lot of actual microbes, living microbes. He termed them animalcules because they looked like tiny animals moving around. His work, Anton van Leeuwenhoek, was not very warmly welcomed by the scientific community at the time because it really challenged everything they knew. But of course, we look back at it warmly because we're excited that, it's the real start of everything, right? Okay. Edward Jenner is attributed as being the first to make and utilize a vaccine, although that's not entirely true. And he didn't know the causative agent. He kind of got lucky. So there's a lot of things we can learn and respect from him, but there's also a long way to go before being able to make more vaccines in the way that we do it now, of course. So he, like I said, is attributed with the first vaccine, which was for smallpox. Smallpox epidemics are very common, and they could be deadly, and they could also just leave someone permanently scarred for life. Okay, so smallpox was almost nearly, it is considered eradicated thanks to vaccines. So this is also a really good example that if you get widespread adoption of a vaccine, and if it is an organism that's only replicating in people, then you do have the potential to eradicate it. So smallpox and polio, right? But polio is not completely eradicated, but it's close. And if we could just, get those vaccines in those remote areas, we could completely eradicate. Okay, so smallpox, a little bit about it. It gives you flu-like symptoms and a rash, but that rash will then turn to fluid-filled blisters, and that can also, like I said, it has a high mortality rate, but if you don't die from the, fever and everything, then these blisters can leave horrible scars. And let's see, the Chinese had actually practiced hundreds of years before Edward Jenner. And we believe this might be where he got the idea from, but we don't know for sure if he had access to this information. But they would use smallpox blisters from people who had very mild symptoms, and they would use that to spread it to people, assuming that they would just get the mild form of the disease. and then they would be safe from the really what we call virulent or super strong, harmful version of the disease. And that worked a lot. It was pretty successful. I think about 2% of people died from this Chinese practice, and that's compared to 20 to 30% of people dying from the outbreak itself. So it was very successful, but it was still risky. 2% died just from this early idea of vaccination. And where we get the word vaccine or vaccination is from vacca, which is the Latin word for cow. And that'll make sense when you get the rest of the story about Edward Jenner. Okay, so Edward Jenner, he made observations that milkmaids who had a similar disease called cowpox never seemed to have a second cowpox illness. And furthermore, they never caught the actual smallpox disease when there was an outbreak. And so he noticed that these milkmaids, they were basically unharmed, untouchable, right? They had the super mild case of cowpox, and then they never had any pox again. And so he wanted to try this. He figured that having that first mild cowpox infection must have been responsible for giving them this protection against further infection. And so he tested it out. Now, I don't approve, and neither does the scientific community approve of the way in which he tested this. We would never just do willy-nilly experiments on humans, right? But there was a different time back then, and that's what he did. So he took a boy, I believe, of one of the milkmaids, and exposed the boy to cowpox blisters. He became, mildly ill from the cowpox, and then he exposed him to cowpox again, and nothing. He was immune. And then he also was immune from actual smallpox. So Edward Jenner, really his biggest contribution is this observation that exposure to a similar weaker version can give you protection from the more deadly form of the illness. But he didn't know that it was a virus. He didn't know smallpox was caused by a virus. He didn't know even about germs, that germs are responsible for disease, right? But he was getting close to the germ theory in a sense that he noticed that the blisters are how you contact the blisters, you get sick, you get blisters yourself, right? So he did make some good observations, and he did figure out how to make this into a form that could be spread throughout the community. And he actually traveled a lot and tried to vaccinate all of Europe, and so you'll read about this in your book. So it's really cool that he was able to help so many people not get the actual smallpox illness. But right, this is not exactly what I would call a vaccine. I mean, it is and it isn't, right? Okay, so great example of the power of observation. It's a morally wrong how he conducted his experiments, but he got lucky. He had great success. All right, so I'm going to actually stop this video here and let there be a third video to finish the history of microbiology.